All right, so first, thanks for the opportunity to speak today. It was a difficult task because I had to recap maybe 100 years of work into this workshop. So I decided to give a simple title to my talk, and basically it is, how can we use replication and repair mechanism to fight cancer? So my first question, I need to give you some questions. So first question, who discovered the double helix of DNA? A, Mendel. B, Watson and Crick. So please raise your hand. Who says Mendel? Who says Watson and Crick? Okay, you're all good. All right. So basically, when we talk about DNA replication, we need to talk about the discovery of double helix by Watson and Crick, and this was done in 1953. And basically, at the beginning, they thought that the DNA was a triple helix, and then they came down to a model where they had a double helix. And by having a model with a double helix, they actually thought that perhaps if we separate the strands, this will lead to a possible copying mechanism, and this is what they found. Why do you need to replicate your DNA? Well, basically, this happens in all our organisms. This is the basis for life. It's a process where two strands are being separated, giving rise to newly synthesized DNA. Unfortunately, DNA replication is used in normal cells, but also in cancer cells. And in cancer cells, the cancer is dividing pretty quickly, so DNA replication occurs very rapidly as well. So this is a very simple view of DNA replication. So here are the two strands of the DNA. So you have each base pair here, adenine that pairs with thymine, and guanine that pairs with cytosine. And basically what you need to do is replicate these strands. So what will happen is, first you're going to have the two strands of the DNA that will be separated, and then you're going to have a DNA synthesis to produce newly synthesized DNA. And this is a process which is called semi-conservative, because each new double stranded DNA contains one old strand and one new strand. So this is pretty basic, right? So to do this, you need an enzyme which is called a DNA polymerase that will add each nucleotide one after the other, giving rise to these newly synthesized DNA. Of course, this is complicated, and you have different strands for DNA replication. So usually it goes only in one direction, from the 5' to 3' direction. So you're going to have a leading strand and also a lagging strand, which uses RNA primers to produce DNA. So this is really the basis of DNA replication. So how can we use these properties to kill cancer cells? But there's different things you can do. First, if you do DNA synthesis, you actually create some super helical structures here, some torsional stress. And there's an enzyme called topoisomerase that relieves actually this stress in front of the fork. So you can use etoposide, camptothecin drugs that will actually inhibit this enzyme. So when you replicate the DNA, this will cause collapsed replication forks and DNA breaks. Secondly, you can use in the clinic different drugs that will halt DNA replication. So one drug that is used in the clinic is called cisplatin. So as you know, cisplatin will bind DNA. It can bind actually on two opposing nucleotides, but also it can actually bridge the two strands of the DNA. So once cancer cells try to replicate their DNA in presence of cisplatin, this causes actually DNA breaks. This can also lead to cell death. Obviously, I think the DNA replication inhibitors, the drugs that are used in the clinic, are pretty old. These are old drugs. And the principle is that the patient will receive a dose in which the normal cell can do DNA replication accurately, while cancer cells, they will get probably mutation and replication for collapse. So that's the basis of one DNA replication therapy. A second one, which is based on DNA replication, is the use of 5-FU. This is also an old drug. And what does 5-FU do? Sorry, can you repeat that? Yeah. So 5-FU is basically, it's an analog that looks very close to thymidine, actually. So that's 5-FU, and that's thymidine. So the structure is quite the same, so it can be actually incorporated in the place of thymidine. So basically you get an analog which is not a base, and this will cause a mutation once you replicate. The second thing that 5-FU can do, it can actually inhibit thymidylate synthase, and this will diminish the level of nucleotide into the cell, also leading to mutations in the DNA. So I think chemotherapy is actually, the chemotherapy based on DNA replication is systemic, and we actually need to use better strategies to fight cancer. So I think basically the better therapies are probably in the second part of my talk, which is really based on DNA repair. This is a new, fairly new field, I would say. Even Watson and Crick actually missed it. So when they've made the double helix, the first thing they thought is how can you replicate this DNA? And they thought that the DNA was very stable because it contains the genetic information. Actually, it's later that they admitted that they totally missed the possible role of DNA repair, and they came to realize that DNA is so precious that probably many distinct mechanisms would exist. Nowadays, we know that DNA repair is very important, and one of the reasons is that your DNA is constantly attacked in cells, in normal cells. So in a single day, the DNA in one cell suffers 20,000 single-strand breaks. Okay, a single-strand break is basically one strand of the DNA which is broken. You can have depurination events where you lose bases, and also you get these double-strand breaks, which are very lethal, very cytotoxic, where you have the two strands of the DNA which are broken. Okay. I think one highlight of the DNA repair field is that I think DNA double-strand break repair has become very important in DNA repair, and for the rest of the talk, I will concentrate on that pathway. So how can you create these double-strand breaks? Obviously, you can create these breaks naturally if you have single-strand breaks in your DNA and you replicate these single-strand breaks. This will cause double-strand breaks. Also, this is the basis for radiation therapy. When you treat cells with ionizing radiation or radiation therapy, you create these double-strand breaks. So the bad news arrive when these double-strand breaks are not repaired. So you can have cell death, and also you can have loss of non-essential chromosome fragments, and sometimes translocations. And this is highlighted here. So these are chromosomes from BRCA2 deficient cells. BRCA2 is important for homologous recombination. We'll see later in the talk. And normally, each chromosome should be one color. This is a chromosome painting experiment. And you can see that something has gone very wrong in these cells. So you can see that some chromosomes have 2 or even 3 or even more colors, suggesting that you have translocations and high level of genomic instability in these cells. Of course, this can lead to apoptosis, but also it can lead to cancer. So the good news is that cells have evolved a way of repairing double-strand breaks, and one pathway is called homologous recombination, and this is a conserved pathway from E. coli up to human cells. And this pathway can repair faithfully double-strand breaks. So and it's quite complicated. So the first step is the double-strand break, which is detected by kinases that will actually stop the cell cycle. Kinases like ATM and ATR. So the goal here is once they detect the double-strand break, you need to stop the cell cycle so you can allow time for DNA repair. Following this, you need to resect this double-strand break into single-strand tails. And this is done with the MR11, RAF50, and BS1 complex, leading to single-strand tail, which can be used by recombinase, such as RAD51, and also mediators, and I'll talk about BRCA2 and PALB2 in a minute, mediators that will actually find the information in an undamaged DNA duplex. And since this process functions only in S phase, where you have a sister chromatid which is present that will give the information for DNA repair. Following this, you have a dissociation, reannealing, and this will complete the repair of double-strand breaks, and you'll have cell cycle restart. So the main enzyme in this pathway is RAD51. And basically, after resection here, this is a recap of what I just said. RAD51 will polymerize as a filament on these strands, and sorry, I'll have to go back. Oh, the video is not working. Oh, it's working. So basically, RAD51 will search for homology, and this will give rise to DNA synthesis to repair the DNA. RAD51 is homolog to RECA, the bacterial enzyme, but there's a striking difference between RECA and RAD51, is that RAD51 is a poorly active protein, and you need mediators that will stimulate its activity. So which mediators stimulate RAD51? That's another question. A, P53, B, PALB2, C, BRCA1, D, BRCA2. So A, who says A? B, P53? Okay. B was PALB2. It's stimulated by PALB2. And then BRCA1, yes, and BRCA2. So basically, this enzyme here that search for homology is stimulated by PALB2 and BRCA2, and also BRCA1. And basically, BRCA2, I think, has been well characterized as an ovarian and breast cancer gene. And in the case of PALB2, this is a less characterized gene. And actually, one highlight, I think, in the DNA repair field is the emergence of this new breast cancer gene called PALB2. So PALB2 is mutated in 0.6 to 2.7% of breast cancer. And I'm being told that once it's 1% or more, this is huge. So PALB2 is really a very important DNA repair gene. And in fact, that's easy for me because we're working on this gene. And there was a paper by Mark Tyszkowicz, and he has done a study with 362 members of 154 families with PALB2 mutations. And he found that if you have a mutation in PALB2, it gives a woman a 48% chance of developing breast cancer by age 70. So this is a fairly new gene, and I think that's one of the things that will be pursued in the future. So what are these proteins doing? I told you that they are mediators, so they stimulate RAD51 for homologous recombination, DNA repair. So basically here what you see is a nucleus of a cell, and you can see that BRCA2 after irradiation forms these little punctuated dots. And these dots represent sites of DNA damage, so they form foci after DNA damage. In the case of PALB2, PALB2 is also able to form foci, but in both cases, if you delete BRCA2 or PALB2, Rad51 is no longer able to form foci after DNA damage. That means that these proteins recruit Rad51 for foci formation and homologous recombination. So if you're not able to do homologous recombination, of course, you're going to get genomic instability and cancer. So that's the next 3 slides are the only slides from my lab. So we're biochemists, and in order to have more insights into these proteins, we actually purify PALB2 and BRCA2 from cells. So here, what we do is that we use a tag, a GST tag that we cleave here, following by a His purification, and this gives rise to very pure PALB2. I think we're the only Canadian group that has purified PALB2 and BRCA2 in a test tube right now. And there's 4 teams in the world that have BRCA2 in a purified form. And here, it took about 4 years to purify this thing, and we have our own ice bucket challenge in the lab. So we were all happy to purify. It's actually 400 kilodalton, this protein. So the future of this, I think, if we think about cancer, is to try to develop drugs that will now inhibit BRCA2 and PALB2, because I don't think there's any drug that can inhibit these enzymes in the clinic. So we actually designed a system where we can actually recapitulate homologous recombination in a test tube. So here we use a label resected DNA and Rad51, and we had an homologous duplex. And if you do recombination, you're going to be able to form this structure called the loop, which will migrate higher on a gel. So you can see here that Rad51 is a poorly active recombinase, as I said previously. And when we had the purified PALB2, we had up to 20-fold stimulation of homologous recombination. So PALB2 is a new breast cancer gene, ovarian cancer gene, and is a very strong modulator of homologous recombination. So now we're in a position to put small molecules and try to see whether we can find molecules that will inhibit PALB2. And I can answer questions about that a bit later. So how can we use these properties, the DNA repair properties, to kill cancer cells? Well, it seemed pretty impossible a few years ago to try to inhibit DNA repair enzymes, because you would probably have the chance to create mutations elsewhere in normal cells. But now it becomes possible. Why does it become possible? That's my question 3. Because of this. So what is this? PARP inhibitors, yeah. So you heard in the conference, I think, about PARP inhibitors, and basically this has changed completely the way we can approach, I would say, therapeutic strategies for cancer treatment. Okay, so how does a PARP inhibitor work? Well, it inhibits this protein called PARP, and this is a protein that will actually use NAD and energy to form these long polymers, and these long polymers serves to bind different proteins. It will bind protein for DNA repair, it will bind protein for cell cycle arrest, it will bind protein for apoptosis. This is a very multifunctional enzyme that works in different pathways as shown here. But foremost, PARP function in DNA repair, and it's able to repair single-strand breaks in the DNA. Okay. So this led to the synthetic lethality concept, and I want to go through this because this is like completely, I think that's a revolution. So the way it works, PARP inhibitor, is that PARP will bind these single-strand breaks and try to repair it. So if you inhibit it, and this single-strand break is actually replicated, this will give rise to double-strand breaks, which in normal cells will be repaired by homologous recombination, as I just said before. But in the case of, that you have a mutation in BRCA2 or even PALB2, you cannot repair these breaks by homologous recombination, so this leads to cell death. Okay? The main advantage here is that normal cells that don't have a mutation in BRCA1, BRCA2, or PALB2, they will remain intact because they can repair their genes by homologous recombination. So of course, this is very efficient. Alan Ashworth has shown that it can actually inhibit ovarian tumor proliferation. That was a paper published in 2010 in Nature. And I think one thing that we should do in the future is actually try to see whether these PARP inhibitors function with other genes. And in the lab, we can see that if you knock down, for instance, PALB2, you have a high sensitivity to PARP inhibitors. So I think one area that needs to be developed is how these PARP inhibitors can function towards different cancer-causing genes. One problem that can arise in the DNA repair field is that you can have resistance to PARP inhibitors, and this can function different ways. For instance, if you upregulate a multi-drug resistant transporter, you can just pump out the PARP inhibitor out of the cell. Or sometimes if you have mutation in homologous recombination genes, you can actually upregulate homologous recombination. And this will also give rise to resistance. So what can you do if you have resistance? Well, of course, you can try to remove these pathways by inhibiting the multi-drug resistant genes, or you can try to upregulate some homologous recombination factors, downregulate some homologous recombination factors. Well, basically, I think one area of future thing is these cancer cells, if they are resistant to PARP inhibitors and they don't have homologous recombination, they always have to find a way to survive. And one such backup pathway that can be activated is the single-strand annealing pathway. And this is actually a pathway which is dependent on the RAF52 enzyme that will repair double-strand breaks through resection and annealing of complementary sequences. So I think in the future, what people will do is that they will use these new RAF52 inhibitors in PARP-resistant cells and try to see whether it creates synthetic lethality. So this is an avenue, I think, that will be developed in the future. And finally, I have like 2-3 slides left. So my fourth question, what is the most important thing? A, the discovery of the iPhone, or the discovery of the CRISPR-Cas? So who says A? A? Oh my God, okay. And who says B? Okay, so you've heard about this system, and this is based also on DNA repair. So what is CRISPR-Cas? Basically, that's a process that was found in bacteria to protect themselves from pathogens. So when phages try to infect bacteria and kill bacteria, there's this enzyme called Cas9 that will actually kill the DNA of the bacteriophage. So the way it works is that the Cas9 binds actually an RNA molecule, which is complementary to your DNA molecule. And this will target Cas9 to create a double-strand break in a specific place in the genome. And basically, you can see that as a Photoshop for genes, because you can inactivate or replace some genes using this process. So basically here, these are the applications you can see with CRISPR-Cas9. So for instance, you will create, when you use Cas9 with a guide RNA, you can target it to genes. In the first thing you can do is that, let's say you have a mutation here in BRCA2, you can create a double-strand break here, and through homologous recombination, you can actually do gene therapy and repair this sequence. A second thing you can do is that there's another mechanism that repair double-strand break called NHEJ. I didn't talk about it. This NHEJ mechanism is error-prone. So once you create the double-strand break, you can actually create deletions in del mutations that will lead to inactivation of a gene. So this might be important, for instance, if you want to do synthetic lethality in cancer cells. So you can actually delete one gene and then put PARP inhibitors and see whether your cancer cell can respond. Right now, this is a very fast-growing field. Okay, Blake Widen-Heft, he's a former postdoc in Jennifer Doudna's lab, the future Nobel Prize. And I have to agree with him. I don't think there's any example in any field moving this fast. We're not at the, I don't think we can cure cancer yet with this technology, but probably if this course is given in a few years, I'm pretty sure somebody will come with a CRISPR-Cas strategy to cure some types of cancer, especially leukemia, which I think is feasible. So I would like to thank the people that helped for this course, which was not easy to do, honestly, because that's quite different than what I do usually. So especially Guy Poirier, he's I would say, the father of PARP in Canada. He worked on these enzymes since the 70s. And also Sylvain Moineau, who was one of the first person to show that Cas9 is able to create a double-strand break in bacteria. And I'll be ready to take any questions.